Hi, I'm Sadashiva Pai from Science Mission. Today we have Dr. Igor Sokolov, professor of Tufts University School of Engineering with us. Dr. Sokolov uh, has an unique background in multiple branches of natural science, high energy physics, quantum field theory, cosmology, gravity, advanced materials, self-assembly, microbiology, microscopy, nanotechnology, nanophysics, nanomedicine, mechanics at micro and nanoscale, economics. Looks like he has done everything. So he has over 150 peer-reviewed publications, including Nature, physical reviews, Nature Nanotechnology, Nature Methods, Materials Today, etc. So he has a scientific citation index of more than 5,500, more than 20 patents and patent applications he has. Welcome to Dr. Sokolov.
Hello.
Thanks for joining Science
Sure.
Hangout.
Well, thank you for your interest and the invitation.
Can you please tell us about your background, education, and some of the projects you are working on?
Well, again, could you remind me how much time do I have to describe all of that?
Maybe a few minutes.
Okay. So I think for the broadness of the background is defined by simply political situations in the countries I was brought up and then had to leave. Because initially I was educated as a high energy physicist, quantum field theory.
Uh-huh.
And that was really interesting because it really answers the questions, fundamental questions of where the universe come from, what is actually built of, and so on. And after collapse of Russian science, after perestroika, I had to pretty much like I had only two choices, either to change my occupation or to change country. And I still liked physics a lot, and I decided, well, let's try. So we immigrated to Canada, where I found first some applications of physics to biology and medicine. And I was really fascinated that it's so much what physicists could do there, because in some applications it looks like the developments around the Stone Age. Some applications and some developments are really stellar, but some are really kind of weird. Of course, there are some explanations why people still prefer to do that simple procedures. But anyway, so that was really interesting. And then I simply consider university work as a sort of exploring of unknown. And everything is interesting. And particularly if it's rewarding, if it's by people recognition, I mean, not in terms of citations or something like that. It's simply recognition that you understand that you're doing things which people need. That's why focus of the interest is in aging, cancer, and recently we also looked at cardiovascular diseases. So that's pretty much the major three problems humankind has, I think.
Okay, tell us about your lab people, like grad students, postdocs, etc.
I have at the moment 4 PhD students and 2 postdocs. One is on leave, though. And I think that's a very reasonable size of the group because I tried several times. Well, it's pretty much ups and downs, so it's very hard to control because support of science in this country is almost impossible to plan. And there is a temptation to develop a bigger group, and so you have more or less like less fluctuations. But if I have more than 5 PhD students and 3 postdocs, what I found, I really spent a lot of time for administration. So pretty much I'm almost like a hostage of this group because I have to write more and more grants to support them. And it's not fun. Because, well, the level of support is such that writing grants is a lot of time counterproductive. But at the same time, I have to say that writing, when you do writing, you better understand something.
Yep.
So that's also what I'm trying to teach, at least postdocs and some students who actually have natural ability to do good writing.
Mm-hmm.
Also to do, I would say, mindful writing, where you write something. It's actually kind of interesting because I think a good writer have to have a sort of bipolar disease. So we have to really have two people in you. One is who is doing writing, and the other guy who is criticizing you.
Uh-huh.
So that's the only recipe how you can do meaningful writing, I think.
Okay. And how do you motivate these people? Like, I mean, like research goes ups and down. There are some good days, bad days, experiments don't work. So how do you keep them motivated, make sure that they work hard?
Yeah, that's well. Technically, I think that they already at that level should be motivated themselves because
Mm-hmm.
in academia, when you work for yourself
Yep.
a lot. Secondly, it's not really motivation, but rather help, because the first thing what I'm keep saying to students when they're ready, that I saw many people in my career who are passed through different stages of their careers. They were initially students, and then I see what happened later to them, what kind of careers they got. And I see one common denominator of all successful people. It's not really intellect or, I mean, well, kind of ability to solve problems or pass tests. It's not even how you verbally communicate. It is one thing. It's called resilience. It's really how you can keep punches or can tolerate downs and failure. And this is the most important part. That's what I'm trying to tell people, that they should really consider failure as a very good educator.
Mm-hmm.
Because if you don't fail, I mean, you cannot really learn anything.
Yep.
So the only thing is like you have to go kind of up and do, at least at a particular topic, you have to do like less and less kind of simple mistakes.
Yep.
So the gradient is important rather than just errors by themselves,
Mm-hmm.
mistakes themselves.
Okay. And can you please talk about some of the role models or the mentors you had in your career?
Regretfully, I cannot really say that I had any a good mentor who could guide me. Maybe because I had to really change my occupation so many times.
Mm-hmm.
Not so many, but like probably two. Uh, but I would say that in terms of motivating mentoring, that was Jeffrey Ozin. He is a professor of chemistry in University of Toronto in Canada.
Mm-hmm.
And it's simply the person who I think was very natural motivator. He has charisma,
Mm-hmm.
and definitely it's one of the people who would be very successful if he decided to be a politician, for example, because he definitely can convince people to do something and feel that they're doing something unique. All this may not necessarily be very important, or kind of even some boring things,
Uh-huh.
but still he projected that kind of feeling of importance, and that's, I think, very important
Mm-hmm.
for good motivation. Because if you think that you're doing a very technical and boring job, so it will not be successful.
Okay.
Unless you have, like, again, like there are some people who actually enjoy doing repetitive work, but I don't think it's really scholars.
Okay. And I know you are busy with all the projects and all those things. So how do you balance your work with your lifestyle? Like, what are your hobbies, interests other than research?
Well, I only like probably hiking
Okay.
from time to time, and I also like to join it because, again, lack of time, jogging.
Yeah. Okay.
Because when you hike, it takes a lot of time. When you jog, it's much faster.
Yeah.
But the amount of relaxation that you get is definitely way more when you jog. Um, and I also like meditations.
Okay.
I think that's very close to how you do experiments or you think about science.
Mm-hmm.
It's almost like, for example, when you learn something, you try to think what you're doing. But when you do something, you don't. It's very dangerous to think.
Mm-hmm.
It's almost like a feeling.
Mm-hmm.
And it's the same like you drive bicycles, so you don't really think where to move your steering wheel, or when you walk, like how to move your legs. So you pretty much like just do that, and that's what ideally the skill that graduate students have to learn.
Okay.
So they have to develop the knowledge. They're so comfortable, they don't think anymore, they feel. And that's pretty much, I think, why meditation I think is good, because the feeling when you do experiments is almost the same. You immerse in something, and you don't really think what you're doing. You just kind of keep doing it and enjoy the process and see something or look for something unusual.
Okay. And coming back to your work, so you are a physicist and all. What got you interested in cells, diseases?
Yeah, in the beginning, I simply was lucky because I immigrated to Canada
Mm-hmm.
in 1994. That was the year of previous year was Ronald Reagan, American president,
Mm-hmm.
closed something super duper, like typical American type of names of projects. I think it's like super conductive, super collider or something like that. And approximately 10,000 energy physicists were out of job. So then I had like negative chances to find a job. So I pretty much look around what I can also do, and I was lucky to be introduced and hired as a postdoc at the Department of Microbiology in the University of Toronto. I simply wrote some equations about their bugs, and they were said, "Wow, you can really kind of present some kind of really scary math to describe our bacteria." So when they hired me to do some sensing work, mostly sensing of DNAs. And that's the first time I was introduced to this area of biology and later medicine. And I was amazed perception of how people treat you around. So I simply felt much more respect. Because if you just do simple uh, physics, people say, "Oh, yes, probably yes, it's very complex things," but it's pretty much not really nobody cares. I mean, cares, but it's kind of one of the interests which are probably stands like 10th or 12th in the line of priorities. But when you speak about medicine, you immediately have some resonance with people, and it's a very rewarding feeling. It's almost like the feeling that you're doing some volunteering job to help people. That's,
Yeah.
I think, what exactly motivates them. And I looked at it, I found like, why all these technologies that we have these days, we still don't understand what cancer is.
Yep.
We still don't understand the interaction of cells when they block arteries of blood vessels. And it's kind of really weird. And we don't understand aging. Aging is really unusual. Like if you look at science fiction, aging is so brain changing or mind changing. So you hardly can find one or two novels, science fiction novels, where humans would be immortal, or at least like live extremely long, because nobody even can imagine what it means. So that's also like looking if it really even makes sense to try to fix human body. Maybe it's like much easier to transfer our consciousness in something like a machine. It's also open question, and that's also very close to my last interest in economics, because again, like I'm from Soviet Union, uh, former Soviet Union, and by all my life there, I proved somebody's theory, and that theory was purely economical, and it turned out it's wrong. And of course, it was very natural for me as a scientist to ask what was wrong. And I checked Adam Smith, first guy who actually initiated the science of economics. And I was really amazed because the guy actually indicated what was wrong there from the beginning. It's simply interest of individual people. For all these economic theories, they ignore, with only maybe one exception quite recent, it's called agent theory, ignore interest of individuals. But I'm sure that's where the whole problems of modern economy is coming, because people, they ignore interest of individual people. They kind of assume that as soon as you work for a corporation, your interests are 100% aligned with corporation interests.
Mm-hmm.
And well, that's what we develop also some theory model which shows what happens if it's not. And it's kind of a lot of interesting conclusions.
Okay. Yeah. So you have done some work on AFM and cell biology. So can you please go ahead and present your work on that?
Yes. AFM is atomic force microscopy. And I was lucky I was introduced to this technology at the time it was born by my PhD advisor. And that time it was theory, and it helped me a lot to converge my interest with medical area, in particular biology, microbiology, because what happened there? My atomic force microscope is, I have like a few slides in the presentations like to explain. It's like a finger. And this finger touching the surface, and you get information about the surface's topography, almost the same like blind person touches the surface and gets a feeling how it look like. And it can work in liquids. You can put your hand under water and or physiological solution, so we can really feel and see cells in their physiological conditions while they're alive. That is why from the beginning there was really big interest from medical area, from biomedical area, in this microscopy. But that time, about 30 years ago, that time people didn't know well forces which work at that scale. Because what you do with your finger, you touch. You don't really touch surface. You touch something which repels your finger. If you put something on your hand, or whatever you would try to touch, you will feel whatever you have on your hand, not the hand itself. Of course, you can push it like harder and so on, but still, it is forces what you feel. That is why this technology is called atomic force microscopy. Atomic is kind of a word showing the ultimate limit of sensitivity of this technique, because it can sense even atomic forces. But the problem was, because people didn't know these forces, you get some recording by a computer, some profile of forces. But what it means, people didn't know well. And it was a lot of artifacts simply produced as a true surface of cells, bacteria, or even molecules. And as a result, biomedical area, scientists from biomedical area were very disappointed in this technique in the beginning. It was even saying that in force microscopy you can produce whatever you like. And it was true, because technically if you don't know what you are measuring, so you get like, as soon as, like even, I remember one of my presentations on the conference, I showed a picture where you have some noise.
Mm-hmm.
And this is a technique that records something. No matter, like, it records force. Do you call this noise or not? It's simply your visual perception. And I see that a bunch of scientists try to convince me that I had to put a graduate students work hard for several months to find one picture would look like better, not this noisy. But the whole point was that this noise was the information we want to analyze, because that was exactly the baby we are trying to grow instead of just throwing it away. And that's why I think main reason why for 30 years this is the only microscopy that hasn't been used in medical area as yet. There was some work done in Europe by one of the founders of this microscopy, but again, it's mostly using sensors developed within force microscopy rather than microscope in itself. That's why, and also after graduation from university, I worked for several years in the Institute of Metrology. And metrology stands for pretty much the same like a local NIST, National Institute of Standards. It's how you do something accurately and precisely. That's why it was my natural desire to make atomic force microscopy as repeatable, as quantitative as possible. And this is what is needed for a medical area. So it should be independent on where measurements were done, by whom, which AFM probe you use, because probes are different. It's like really very sharp needle, which you can buy commercially from different manufacturers, and so on and so on. That's why cells were always the interest. This microscopy has unprecedented resolution. It can give you X, Y, and Z, like in all three directions resolution. It can give you maps of physical properties of surface of cells, again, with resolution that no other technique can give you. So it was always on the table that this microscopy could definitely give us some knowledge which no other techniques could give you.
Okay. Can we learn from your work? Like, can you present your work so that we can learn more about?
Absolutely, huh? So let us. I have a presentation which I think I also added some recent work, not about force microscopy, but also about nanoparticles. And it relates to force microscopy because here we can see with recent development in force microscopy, we created two new imaging modes in my lab. And this one of the modes allows you to see surface coating on nanoparticles.
Mm-hmm.
And this is, I think, crucial for what we call nanomedicine these days. So this is pretty much the presentation, and I will. So do you see it?
Yeah.
Uh-huh.
Go ahead.
So on the background, you see a typical picture of a cell imaged by atomic force microscope. It's in 3D. And because what force microscope records, it's a position of, let's say, height at each point or location of the surface. And it's recorded by a computer and can be presented pretty much in any color, any type of presentation of digital image. So nowadays it's like almost unlimited possibilities. So that's the outline of my presentation. I want to tell you about ultra-bright fluorescent nanoparticles for cancer detection and drug delivery. And that's where, again, atomic force microscopy is the key. And second, atomic force microscopy is our recent work as non-invasive method of detection of cancer, and particularly shown how it works on bladder cancer. That's also our recent work, just published. I think it's not even officially published. It's just online. And that's number two. It's, I think, could be the first clinical application, if it's adopted, of this microscopy.
Mm-hmm.
By the way, if you feel that something is not clear, so just interrupt and
Okay.
questions. So, well, first, what is nano? It's more or less like trivial. These days, probably everybody can look at it, and this is Wikipedia slide showing that nano is something. I don't know. Do you see my mouse?
Yeah.
So, so it's something between molecules, small molecules and atoms, and large objects like, well, in this case, it's genes, but genes are quite large. You can see them under even like good magnifier. You don't really need a microscope for that. So some bacteria cells, some dust particles. If you go to a desert, so you can get some particles of the size of like several microns in the air. Human hair, for example, is about like 10 microns. And 1 micron is 1,000 nanometers. So What is the problem of nanomedicine these days? Nanoparticles are really beautiful objects for targeted drug delivery. And cancer was very natural choice because typically cancer is treated with chemotherapeutic drugs, which are simply some toxins. And the only difference between these toxins and any other toxin, that this toxin is more toxic for cancer cells than for normal cells. But still, it is toxic to normal cells. And that is why there is a big desire to deliver these toxic drugs only to tumors instead of just spreading to the entire organism, how it's done right now. And that was on the table for quite a while. Picture here is from 2005. It's a number of papers. Pretty much, this is number of papers using different, describing different nanoparticles delivery. And here it's efficiency in percents. Efficiency, I mean, how much you, for example, you put, let's say, 1 gram, and this is what the percent of that 1 gram is delivered to actual tumor. So, and this is presented by years, like delivery efficiency in percent for years. And you see that in median, and actually looking at this, there is no progress for the last 10 years. So we try to think to understand why. I think, again, our hypothesis that it based on the same issue that force microscopy faced before. People don't quite understand forces. Forces acting at that scale between nanoparticles and the surrounding of the organism, which nanoparticles face. And why forces? Well, because like imagine that you are this small nanoparticle. You have to really get sticky to only cancer cells, but you're in a very complex environment. And because of this very low delivery efficiency, it means that you stick somewhere, not in the appropriate place. But stick, it means there is an attraction force that you attracted to something which you're not supposed to be attracted. As a result, the efficiency is low. Now, how we try to understand with the help of force microscopy. So this is pretty much how force microscope works. There is some small chip made of silicon with a cantilever, like this V-shaped cantilever, with a very sharp probe or needle attached to the apex of this end of this cantilever. Cantilever is very flexible. It can go up and down. And deflection of this cantilever is detected by a laser light, which is deflected from the top and detected by some photodetector. Let's see this guy. Can you see it moving?
No.
Ah, okay.
A movie doesn't work most of the times. Yeah, sometimes.
So I think that's because I do see it on my screen.
Oh, okay.
So it's a problem of sharing.
Yeah.
But in any case, like if there is a bump, and at the same time this surface of a sample moves left or right, this bump would push this needle up, and the needle pushes the cantilever, so the cantilever deflect up and down, and the angle of reflection of laser light changes. That's the basic mechanism of detection of signal in force microscopy. So it's a deflection of the cantilever. And the cantilever is like a small spring. The bigger detection, the bigger force. Linear proportionality. So what we recently developed, it is a new mode of force microscopy. By the way, this was it was kind of irony because this mode, we call this ringing mode, was commercialized almost simultaneously with its publication. So university got a patent for this mode faster than we produce a paper for publication. This is like the first time in my career, and I never heard about these things. So it was, and this mode allows you to see 8 additional channels, almost like 8 dimensions of a surface. And one of these channel is the height of the neck. For example, when a probe touches the surface, it's flexible, and there are some molecules coating the surface. And then probe disconnects from the surface. Probe can go up and down. When probe disconnects from the surface, it pulls small neck from the surface before it disconnects. So one of the channels is we measure height of this neck. And the probe goes at one point of the surface, then it goes to another point, and so on, and creating the entire map of the surface. So the second channel is disconnection with the disconnection distance. It's the size of the molecules which are covering the surface, because a probe pulls these molecules out of the surface due to some non-specific interactions. And when it does it, it loses energy. The cantilever loses energy, and using some math, we can restore how much energy it lost, and we can restore the height of these molecules. This is a picture as an example of a surface of a cell, but what you see here, it's pretty much like by colors. Like it's a more reddish, it's a higher. Like blue, it means it's deeper. So this is distribution of molecular coating on surface of a cell, and this is it's 2 by 2 microns. So there is no other technique which is capable of getting such information. So what you see is presumably distribution of glycocalyx. So it's like so fresh picture, so we don't even know exactly what it is. But again, by the size, it's molecular coating can be only molecular coating on the cell surface, which is called glycocalyx. So we use this technique to see molecular coating surrounding nanoparticles. Because remember I said that nanoparticles should stick only to tumors, but somehow they stick everywhere else, but only like 1% at best reaches tumor and stick to tumor. So coating, so these particles are coated probably not sufficiently. So coating can protect them from sticking to everywhere, like small kind of a brush pushing away these particles from everywhere. But it's maybe not enough. And this is a picture example of what we see with this technology. So this is a picture of simply on the left, it's height image. The brighter is higher. And but on the right, you see that it's a coating. And the coating, it's not homogeneous. You can see, for example, here you have less brighter area, which means it's depth. It's a deep or kind of, or even like no coating at all. And there is a pretty big coating here. So that's where we started to differentiate particles by the height of the molecular coating and see, do we have defects on the surface or not? And secondly, but this might be a little bit, and this is where we actually stand right now. We are now trying to see the targeting of tumor using nanoparticles which have different molecular coating, with and without defects. We were lucky to develop some particles without defects. And some preliminary experiments already show that they work very well. And to see the tumor, we developed particles which we called ultra bright. They are substantially brighter than quantum dots. Quantum dots are, at this point, it's like small semiconductor crystals or polymer particles, also semiconductor, which are very bright. So compared to classical quantum dots, we have maybe 10 up to 800 times brighter. And if you compare to the very accurate if you compare to so-called polymer dots, which are polymer semiconductive particles. We have maybe like 2 times higher, but our particles are silica-based. And quantum dots, if they're polymer or not polymer, there are still a resolved problem of toxicity. So you need a brightness of fluorescence. By the way, for those who might not necessarily know what it is, fluorescence, you use one light, but you shine one light, but the surface or particle shine you back a light of different color. Why it's important, you always can put a filter and remove this excitation light, but only see the light of different color, which is coming from particles. And this is a huge advantage because you can see very, very small sources of light. It's almost like you put some filter blinding light around, you see only like night, dark night, and you see only stars. Stars are the particles which are producing light. So this is a technology which allows, for example, to see tumors for a surgeon or for a doctor who does diagnosis. You need to really see some particular lesions or abnormality in your organism, and fluorescence works the best for that. So this is an example of zebrafish. In red, maybe here it's a little bit better. So green, and here a particular like whitish, it's collection of very bright spots where we have a lot of particles. And here in the red channel, we have tumors. It's melanoma in, oops, sorry, it's not melanoma. It is cervical cancer. Human cervical cancer introduced into zebrafish. And you can see a very nice colocalization. So our particles reach those tumor sites and accumulate it in them. So you can kind of look at the same position on fish and see that particles which are this greenish are locating around this, not melanoma, but cervical cancer sites. So, and maybe we can skip it. Why we need? Yeah, that's by the way one very important part. We also develop particles with the help of also force microscopy, where we can see good coating, and it's colorectal cancer. This is a regular colon of a mice, this mouse, and this is a healthy part, and here you have tumors developed. And what we did here, we simply rinse this colon with a solution of our particles and wash them away. Within 15 minutes, we get a very high contrast. So contrast, we see glowing size of tumor. So if you look kind of high resolution, so you can see like a lot of color coming from tumor, and this dark picture means like healthy tissue. The mechanism of this targeting is not 100% clear, but obviously this might be very helpful for detection of small lesions during colonoscopy, because colonoscopy is a technique to check if a person may have some colon cancer. It's kind of visual inspection of colon, but small lesions can be easily missed. And this is the way where it could be helpful for doctors. Also, if particles can accumulate there with fluorescent dye, because particles are fluorescent because we charge them, they're porous, we charge them with fluorescent dye. But we can charge them with, we can load them with anti-cancer drugs, and then it would be an interesting delivery vehicle for cancer treatment. So that's where force microscopy and quite recent development help us. So that's maybe like a little bit of why we think, I think like for the sake of time, I can skip this. It's the interesting part about a cell that and a force microscopy, that you literally can feel molecular coating around cells. Picture here, it's schematics of how cell look like. You have the cell body, which may be wrinkled, and then some molecules surrounding cells. Pretty much all cells, besides maybe a very particular part of bacterial cells, all cells have this molecular coat. It's a very natural protection against invasion of some bacteria, viruses. And if you try to do regular force microscopy, you don't really see these molecules because they are too soft. Sharp probe would simply deform them. Instead, what you do, you can simply put a, I don't know, some sphere, for example, spherical probe, which are bigger. You don't have that high resolution, but then when you start to press this cell, you start to press a lot of these molecules and you start to feel them. And then that's where force microscopy can help, and we can distinguish distribution of these molecules. And it turned out it's kind of some math. And this is an example of this spherical probe. And through some math, we can derive the number of these molecules on cancer cells versus normal cells. And this is what we are kind of trying to do now. We do see that cancer cells have substantially different molecular coating around them. These are non-specific molecules, mostly molecules of polysaccharides, which people don't know what exactly the role of these polysaccharides. There was some work, they were thinking about that these polysaccharides may help to avoid the eye watch of immune system. For cancer cells are kind of hiding from the immune system, because if immune system see cancer, is doomed. But somehow they try to avoid it, and there are like multiple ways to do that. And one of the ways, people think they produce too much of these hiding molecules around them. But also what we found that it's not only that. This structure of coating, molecular coating cancer cells, is much more sophisticated, kind of rougher than coating on normal cells. And that's where we come towards direct imaging of such cells. And the idea, again, came from exactly what I just said. We do see molecular coating on cancer cells to be quite different from normal cells. So how to do that? We found that difference in the molecular coating on cells extracted from human cervix, human cervical cancer. But when I came to doctors just to ask, do you have some samples? We really would be glad to analyze them. They said, why do you want to look at cervical cancer? In the United States, this cancer almost eradicated. It's mostly kind of still you can find some more cases in South Texas, and there is a strong center there. And on the East Coast, we can offer you much more interesting, like in quotation mark, of course, interesting task. It's bladder cancer. Bladder cancer, like different statistics gives you a little bit different numbers, like third or fourth cancer responsible to mortality. It's third for men. It's less prevalent in women. But what is interesting about this cancer, it is the most expensive cancer to monitor and treat per patient. And it's also, in terms of suffering, probably one of the worst, because the procedure right now, as soon as the cancer is identified, you have to be watched, or even if it's treated, you have to be watched between 3 to 6 months with a method, clinical test, which is called cytoscopy. Cytoscopy is a really invasive method. It's like there is a video camera which put through your genitalia, so to visually examine bladder, surface of bladder. And it's not only painful during procedure, you have anesthesia, but later you can get pain like up to 1 week, like what people told me. So it's something which people try really to avoid, but so it's pretty painful examination, which you have to really do for quite a while. So there is definitely people trying to find some methods which would be less invasive or maybe non-invasive at all. And what we tried to do, it turned out, which actually was a news to me, that our urine contains cells exfoliated from the surface of bladder and other parts. When we do urination, we expel some cells. And to collect these cells, it's quite easy, and hospitals already do that. But what they do so far, they simply examine these cells under optical microscope. And of course, optical microscopy has very limited resolution. And this technique is definitely not sufficiently accurate and not really treated as a serious alternative to cytoscopy. So, of course, like what we try to do is to apply our methods to scan these cells. And that's what we, that's pretty much, like I said, that our new ringing mode allows to image multiple channels simultaneously. And this is just, again, a picture, surface of cell. This is melanoma cell, actually. This is height, bright is higher. And there are multiple channels like energy losses channels, this height pulling of neck, and disconnection distance, showing you a distribution of molecules on the surface of such cell. So you can see there's quite a wealth of information. All of that is not a qualitative picture. All of them are really in nanometers or in kiloelectron volts. So all of them are independent on, well, at least we check it, and within a broad range of imaging parameters, they are independent on the type, the way you do the experiments, the way you do the imaging. And this is super important because it has to be repetitive. So what we did, well, we had 43 healthy patients, and actually healthy, it's not a good word, because more than half, I think even more than 2/3 of these people had already cancer, bladder cancer before, and they're in remission right now. They don't have active cancer. And 22, actually right now we have already 25 cancer patients which have active cancer. And that was defined by simply clinical dissection. So they were taken pieces of tumor from their bladder, and after histology, it was clinically identified that those guys have cancer. So, and like this kind of, I skipped some technicalities. So essentially we scanned the surface of these cells. And by some historical reason, we looked only at one channel, adhesion channel. Not all these 8 channels additional that I said, it's simply adhesion channels. Simply because we started collecting these samples, that time we didn't have our new mode as yet. So we keep collecting, but we can at this moment have enough statistics only for the adhesion channel. Of course, height. But height, we saw that height image actually doesn't give you very good information. Adhesion channel, it does. And previously, that's what I said about epithelial cervical cells, carcinoma, cervical carcinoma. We did answer the question of if cancer can be treated as chaotic behavior or not. It's simply if doctors can look at cross sections of tumors this is almost, I would say, first year medical students. You can easily identify tumors because it looks like chaotic. That was a thought that on the level of cells, cancer also would be chaotic. But what we found, we looked at this model of cervical cancer starting from normal to immortalized cells, which are considered to be precancerous, towards cancerous. And we found that it's the stage of immortal is already changed, and we analyzed so-called fractal dimension. Fractals are, it's a geometry, property of a geometry, which typically occur during some chaotic processes. And that's where why we looked at this and tried to understand, do we see some fractal behavior? And oops, I really don't have that slide here, sorry. What I actually found that cancer is not chaos, it's not fractal. It's only, we observe this pure fractal only at the stage between transition, between immortal to cancer. Immortal, you can think about this, like it's a benign tumor. It's really growing, but it's really not dangerous as yet. But cancer is malignant. It's like where you can get some invasiveness. So that we don't quite understand why it happens. So it's definitely we need to find some biochemical way. And why it's needed, why you would say, like, who cares in some sense. But because as soon as we know if it's really due to some chaotic behavior in biochemical reactions, chaos is typically identified through, I mean, it can be influenced through some specific points of bifurcation. So there is kind of a chain of chemical reactions. And in that chain, there are some points which are substantially more important than others to produce chaotic behavior. So if you find, try to find those points, we can definitely influence this behavior. And who knows, maybe cancer. It's of course like highly speculative, but at least it's something which people haven't really thought before. So it might be a new way to attack on cancer. And the last thing is when you see different images, of course, it's always the question, like, can you really identify using, for example, some machine learning? Google, Facebook now can easily identify our faces, like even like iPad, iPhone. But can we really do something similar to these images? Potentially, probably yes. But it would bring us to the problem of so-called big data. One of the problems of big data, you need a big database to train your machine learning algorithm to identify, to teach this algorithm who is who. And we don't have that luxury. We have already like 43 normal and 25 cancer patients. And that took us like more than 5 years because of, like, when you do something new, you don't have money for that. I mean, that's quite regretfully, it's very hard to get support for something truly new. So that's why it was on pure enthusiasm, and it took like quite a lot of time. So we simply, if you really treat this image as a picture, we would need many thousands of patients before we actually can make the training. So that was that end. So what we did, and it's known in this type of problems, we try to reduce number of information per picture. So instead of using the picture, so we reduce it to number of so-called surface parameters. For example, like roughness, directionality, fractal properties, and so on. Those parameters is what people use to characterize surface in engineering. It's a known list of approximately like 40 parameters. That's what we did. And then we tried different tests, including neural network. Neural network actually was not very good because it's so easy to overtrain to produce some kind of false results. And so that's we build so-called learning trees. Learning trees, you have like say like one parameter, you choose some threshold and say that below this threshold you have like good indication that it's cancerous, below it's healthy, and so on. And you kind of try to build sort of a tree which ended up in definitive that this is cancer, this is normal, this is cancer normal, and so on. And there are like different methods. It would definitely be a separate lecture about how these methods work and why we did it. But what do we need? Accuracy. So this is just a picture showing that some parameters are more important. So the bigger bar is the more important parameter for segregation between cancer and normal. And you see that height is kind of like all parameters almost good. But in the adhesion, there are kind of very pronounced number of parameters which are telling us what is cancerous, what is not. And we did some statistics using only one cell. And again, like for those who don't really know statistics, it's just look at this graph. It's accuracy. It's about 80%. This is number of surface parameter we take into account. And you see that around like 10, it's already like good. So we need 10 surface parameters to do prediction. If you use 5 cells, because we can combine different cells together to make a better prediction. Even one cell is like 80% accuracy. It's one cell, and person can make a mistake when analyzing one cell. It's definitely much safer when you do analysis of 5 cells. And when you look at it, you already have more than 90%. To be precise, it's 94% accuracy. And if you compare with the presently existing method, it's around like 80% what we have. So, of course, like you also have to say like how significant this difference, because you have a very limited number of people you analyzed. There is a way how you unambiguously answer that question. It's called statistical significance. And we stopped our analysis where our enthusiasm and money actually ran away. Actually, was no money from the beginning anyway, so just mostly enthusiasm. And we said we had to publish it now. It's exactly the point where we reached a number of people where we do have statistical significance. Our method as it is already statistically significantly better than this invasive cytoscopy. So that's, oh, by the way, this is also kind of important. You always can check if your machine learning method is good or not in terms of overtraining. Because if you do like you train, it gets better, better, and better, but only for that database that you do the training. If you choose something new, your method can fail. And that is the biggest problem of neural networks. That's why we kind of step away from it. But how to check? Do you have overtraining or not? You take your database in which you have these surface parameters for each person and clinical diagnosis: cancer, healthy, cancer, healthy. And then instead of cancer, healthy, you put a flip a coin, 50-50. You randomize it. So randomized diagnosis. And then you run the same procedure, develop your algorithm to check its accuracy. And what we saw, our accuracy was 54%. Before it was 94. This is like 5 cells. So obviously we do have pretty much what we expect, close to 50%. So we don't have any overtraining in our algorithm. That's important. So that's pretty much it. So that's where I think the most exciting developments. We really want to produce something with force microscopy which might be directly applicable to real life, to medical area. Of course, I removed a lot of technical details showing that we do have a lot of verifications that our method is independent on the way we do analysis, we do the imaging. So it is truly robust. So the paper about this last results will be published in Proceedings of National Academy of Sciences. And the paper is, I think, like 3, maybe like 4 pages long. And I think there are more than like 30 pages of supplementary materials showing exactly like what I said. We do have a very reliable method. So it's truly quantitative, and I hope that we can get money from National Institute of Health to do the final study, polishing the method to answer one more question. Can we really distinguish not only presence of cancer by the stage of development of cancer? Because that's what doctors need a lot, because it would define the way they prescribe the treatment. And this is important for it's a life-saving decision. So that's pretty much it. And like, if you have any questions,
Yeah, can you please unshare it? I have one. Like we are almost out of time. Just I want to just
sure.
Like how long does it take for the AFM rather than the regular screening and all those things they do? Like what is the time? Like if you get the samples today, how does it happen within a day? Like what is the time period?
A very good question. That's one of the reasons why we developed this new mode, because the previous relatively standard method we started, it was painfully slow. For example, previously for one cell, it took 1.5 hours to get a picture of one cell, and you need at least 5 cells. With new method, we have like test done within 1.5 hours, the whole test. In principle, we are working now, and we do have already it's developed, but not really data as yet from patients. We do have the test even 4 times faster. So definitely within 1 hour, we can get the answer. And that's what typically doctors said will be competitive. Because right now they do have some alternative tests using mostly biomarkers, which are not as accurate. But it requires about like 1 hour. That's a requirement.
Other question is, this is the last one. So do you think the surface, like you have a different polysaccharides on the surface with the cancer, and as it progresses, and can you catch it using AFM?
The particular type of polysaccharides, probably no. Although in principle, yes, but you need really to attach some sensitive molecules to the AFM probe, and it might be a little bit of clinically difficult, because you can contaminate the probe. So, for example, another question would be like how long you probe, because you touch surface, you can contaminate the probe. For how long our probe would last? In our case, we easily last like more than 100 cells with no changes. And we do preconditioning of the probe a little bit, so you need to touch several times, so it creates a very repeatable structure. So answering your question about type of polysaccharides, we do have kind of research type of methods, but such probe would be contaminated too fast to be clinically applicable.
Okay.
But technically, I would say that it's probably better to use some fluorescent nanoparticles or some more classical methods. It's actually such methodology exists. It's called glycomics, where people try to analyze coating on cell surface and see like what type of polysaccharides they have. But you need a lot of cells for that. So at this point.
Yeah. Okay. Thank you so much. I really appreciate your time. It was very nice. Like I'm not a physicist, I'm a biologist, so I learned a lot from what you have mentioned today, and certainly wish you good luck in your future work. And if you think there is something different, something new, you can always get back to us. We can have another one sometime.
Thank you. Thank you very much.
Thank you so much.
And again, I don't know how you do that, but if there are any questions, so I'll be very glad to answer them.
Yeah.
I think it's very important. Like,
sure. So what I'll do is I'll put it on Facebook, LinkedIn, everywhere. So if at all, I'll give you an email too. If anybody has any questions, they can directly contact you.
Super. Thank you very much, and have a good one. Happy holidays.